Welcome to the deep dive. Today, we're diving into something really fascinating, maybe a plant you think you know. The Venus flytrap, Diania muscipula.
It definitely has that B-movie monster image, doesn't it?
Totally. But the science behind it, well, it's incredible. The sources we looked at show it's this amazing biological machine. It's like an electrical counter and a super efficient recycler all in one.
Exactly. A lot more sophisticated than just a snapping leaf.
Right. So our mission today is to really get into the physics and the chemistry that makes that famous snap happen. Okay, let's unpack this.
Where should we start? The trigger.
Yeah, let's start there. It all comes down to those little trigger hairs inside the trap, that leaf structure. It's a snap trap, sure, but it's smart.
Mm-hmm. Smart how?
Well, it needs two separate touches, and they have to be pretty close together within about 20 seconds or so.
Ah, right. So it doesn't just snap shut if, say, a raindrop hits it or the wind blows some debris in.
Exactly. It avoids those false alarms, saves its energy for an actual meal.
And when it does snap, it's incredibly fast. We're talking about a tenth of a second.
Whoa, 0.1 seconds. That's wild for a plant. How is it like muscle?
Well, that's the surprising part. It's not muscle at all. It's driven by electrical signals, action potentials, or APs, as they're called.
Action potentials, like in our nerves.
Sort of, yeah. The plant version.
Ah.
These APs cause a really rapid shift in water pressure inside the leaf cells, turgor pressure.
Okay, so it's water pressure, but moving that fast. How does the electrical signal make water move quick enough for a 0.1 second snap?
It's about triggering a sudden change in the leaf shape. The electrical signal makes ions move, water follows super fast, and the whole structure flips its curvature.
Yeah.
Think like popping a soft contact lens inside out. It goes from convex to concave almost instantly.
Geometry powered by electricity. I like that.
Mm-hmm.
Okay, so the APs snap the trap shut. But that's not all they do, is it? The counting continues.
Precisely. This is where it gets really interesting. After the capture, the trap doesn't just sit there.
Right. The struggling insect keeps bumping those trigger hairs.
Exactly. And each bump generates more APs.
The plant is essentially, well, counting these signals after the trap closes.
To make sure it's worth digesting, like confirming it's actually got something nutritious.
You got it. It's an energy management thing. It needs about 5 APs total. The initial 2 plus more from the struggling to really kickstart the production of the digestive enzymes.
The hydrolases and things.
Yeah, the hydrolases, hydrochloric acid, the whole digestive cocktail. It's costly stuff for the plant to make, so it won't bother unless it's pretty sure of a payoff. If the bug escapes quickly, no big enzyme investment.
That makes sense. Smart energy use. Now, about that digestion. Most plants absorb basic nutrients, right? But the flytrap does something different, something faster.
Oh, absolutely. It takes a huge biochemical shortcut. It uses something called endocytotic uptake.
Endocytotic uptake. Sounds complex. What's the advantage? Well, instead of breaking everything down to the simplest building blocks and then using energy, ATP, to rebuild what it needs, it just absorbs bigger pieces directly. Think whole amino acids, larger molecules straight from the insect soup.
So it's like grabbing ready-made ingredients instead of starting from scratch.
Exactly. A massive energy saver. And it seems particularly good at grabbing ammonium, which gives it that vital nitrogen it needs, especially living in those really nutrient-poor, boggy soils.
Okay, that covers the organic stuff. But what about minerals, like salt? Do the APs play a role there too?
They do, actually. The number of APs that count also controls how the plant handles sodium. It manages the expression of a specific sodium channel gene, DMHKT1.
Why sodium? I thought plants needed potassium more.
They usually do, but flytrap habitats are often low in potassium, so it seems the plant efficiently grabs sodium from its prey and uses it as a kind of osmotic substitute. Helps keep the cells rigid, maintain that turgor pressure we talked about. It's like using cheap salt when the expensive stuff is hard to find.
Wow. It's managing electricity, chemistry, even mineral substitutes, all triggered by touch. Connecting this to evolution, then, did it have to invent all these tricks from scratch?
And that's maybe the most elegant part. No, it didn't. The molecular studies show it repurposed existing systems.
Repurposed? Like what?
Primarily its defense mechanisms. The genes and pathways it uses for digestion are remarkably similar to those other plants used to defend themselves against, say, being eaten by herbivores.
So the systems meant to fend off attackers were kind of flipped, turned into tools for attacking and digesting.
Exactly. It turned its shield into a sword, basically. A key player here is a hormone called jasmonic acid, JA.
JA. I've heard of that. Isn't that usually triggered by wounding in plants?
Precisely. When a normal plant gets wounded, it ramps up JA production for defense. In the flytrap, the insect triggering the trap hairs, that simulates wounding.
Ah, okay. So the JA levels shoot up.
Right. But instead of just defense, that JA signal tells the glands in the trap, okay, time to start digesting. And yet what's wild is when they looked at the genes, the glands actually look a lot like roots, genetically speaking, in how they grab nutrients.
It just adapted that ancient defense response for feeding. Incredible recycling. So what does this all mean, putting it together?
Well, it means the Venus flytrap is so much more than just a mechanical trap. It's this highly sophisticated biological circuit.
Managing electrical counts, triggering targeted chemistry, repurposing old genes. It's an optimization masterclass.
And it's powerful, too. That trap clamps down hard. The force needed to escape during full digestion can be up to 4 newtons. Imagine.
That's quite a grip.
It is. But despite all this incredible biological engineering, its situation in the wild is, well, precarious.
How so?
Wild populations have plummeted, down over 93% since the late 70s. Mostly habitat loss, also poaching. It's officially listed as vulnerable.
That's terrible. Such a unique organism facing such decline. It really makes you think. This plant's ability to count signals to repurpose defense into offense, it makes you wonder, doesn't it?
Wonder what?
Well, what other amazing biological shortcuts, what potentials derived from existing genes might other plants, the non-carnivorous ones, be overlooking right now in their own genetic toolkit? What efficiencies are waiting to be unlocked?